everyone, welcome back to Biophilia, your all-time study mate. Today we are going to continue to understand about the chapter Cell Cycle and Cell Division with its part 3 about meiosis. If you like our videos and want us to help out reach out many more, then don't forget to like, share and subscribe to our amazing channel Biophilia. So let's continue with the last part of this chapter. हमने इसके पहले के दो पार्ट्स में इन द फर्स्ट पार्ट वी हैड सीन अबाउट द टॉपिक ऑफ सेल साइकिल, जहाँ पे हमने पढ़ा था अबाउट इंटरफेज एंड देन वी ऑल्सो सॉ अबाउट अनदर फेज दैट इज कॉल्ड एज एम फेज. एम फेज में दो चीज़ें थी, दैट इज स्प्लिटिंग ऑफ द न्यूक्लियस एज वेल एज स्प्लिटिंग ऑफ द साइटोप्लाज्म. स्प्लिटिंग ऑफ द न्यूक्लियस में वी हैड टू पार्ट्स, जहाँ पे वी सॉ अबाउट द माइटोसिस, जो माइटोसिस कवर हुआ था इन द पार्ट 2, एंड सेकेंड होता है विद द हेल्प ऑफ मियोसिस, जिसको हम पढ़ रहे हैं राइट नाउ इन द पार्ट 3. सो सबके लिंक्स आपको मिल जाएंगे इन द डिस्क्रिप्शन बॉक्स एंड दैट वुड हेल्प यू टू अंडरस्टैंड ऑल द कॉन्सेप्ट्स इन वंस अगेन मच मोर डिटेल over here on Biophilia. So let's continue understanding about meiosis. अब जैसे माइटोसिस अगर आपने पढ़ा है फ्रॉम दिस वीडियो देन यू वेरी वेल नो कि माइटोसिस जो होता है इट इज़ गोइंग टू ऑकर एंड इट इज़ ऑल्सो कॉल्ड एज इक्वेटोरियल और सोमैटिक सेल डिवीजन. तो अभी माइटोसिस से मियोसिस इज हाउ डिफरेंट और व्हाई इज इट कंसिडर्ड एज अ डिफरेंट टाइप ऑफ न्यूक्लियर डिवीजन, उसको समझने के लिए आपको माइटोसिस की फुल थ्योरी पता होनी चाहिए. यू शुड नो द डायग्राम्स सो दैट यू कैन अंडरस्टैंड द डिफरेंस बिटवीन द टू डिविजन्स. So let's start about the meiosis. So meiosis जो होता है it is going to be a double division. Why is it a double division? You will very well understand because जब एक सेल होता है which is going to be diploid in condition, diploid condition से first meiosis 1 होगा, meiosis 1 से 2 cells बनेंगे. These are going to be haploid in condition and again these cells are going to divide, that is meiosis 2, और 4 haploid cells फॉर्म होंगे. सो फाइनली व्हाट इज गोइंग टू हैपन, द मदर सेल विच इज गोइंग टू बी डिप्लॉयड, ये डिप्लॉयड कंडीशन से फाइनली जो डॉटर सेल्स होंगे वो डॉटर सेल्स यहाँ पे कितने आएंगे, डॉटर सेल्स आएंगे 4 विच आर गोइंग टू बी हैप्लॉयड. सो दिस इज अ वेरी इंपॉर्टेंट डिफरेंस दैट हैपन्स इन मियोसिस. जबकि माइटोसिस जो होता था, माइटोसिस में देयर वाज वन सेल विच वाज डिप्लॉयड और माइटोसिस होने के बाद 2 डॉटर सेल्स फॉर्म होते थे विच वर ऑल्सो डिप्लॉयड. सो द जेनेटिक कॉम्पोजिशन यूज टू रिमेन सेम इन नंबर, whereas in meiosis because of double division of the diploid cell there is formation of 4 haploid cells and the number of chromosomes are going to reduce to half. So mitosis का difference और meiosis का difference needs to be understood over here, that is these are going to form 4 haploid cells and even the number of chromosomes will be half. So that is first major difference that is going to occur between the two types of divisions. Second we have to understand कि since it is a double division वहाँ पे 2 sequential cycles form होंगे, but the sequence 2 divisions of nucleus, whereas the cycle of DNA replication will be only once. So this is very important. जैसे माइटोसिस में एक डिवीजन होता था और एक बार सेल का DNA रेप्लिकेट होता था ड्यूरिंग द एस फेज, राइट. एस फेज एक बार होता था, एक बार मियोसिस हो गया, 2 डॉटर सेल्स आर फॉर्म and the division is complete. But in meiosis there will be once DNA replication in the S phase, but there will be splitting of nucleus twice, जिसकी वजह से the number of chromosomes will reduce to half. So finally what is the use or what is the purpose of undergoing meiosis? So the cells which are going to be requiring to produce haploid cells, सो जो सेल्स डिप्लॉयड है, डिप्लॉयड सेल्स है, जब उन्हें हैप्लॉयड में कन्वर्ट होना है दे नीड टू अंडरगो द प्रोसेस ऑफ मियोसिस सो दैट दे कैन प्रोड्यूस द हैप्लॉयड फेज इन द लाइफ साइकिल. सो जो जनरली सेक्सुअली रिप्रोड्यूसिंग ऑर्गेनिजम्स होते हैं उनको अपनी जेनेटिक स्टेबिलिटी के लिए दे नीड टू रिस्टोर द डिप्लॉयड फेज. So very importantly I can tell you one example. For example, if you have a female which is going to have a cell that is in 2n condition, और अगर उसने 2n condition से ही अपने gametes बनाए and this is going to be her egg, right? वैसे ही मेल का जो है, मेल के पास भी 2n सेल है, he also produced the sperm which is 2n. अब जब egg और ये sperm fuse होंगे what is going to happen? 2n and 2n is going to give you the organism which is going to be a 4n. But क्या हुआ, parents जो थे, parents are going to be 2n and both are there in the diploid phase. But अगर उनका daughter cell is going to become 4n, that is not going to make the species same. तो इस कंडीशन की वजह से whenever there is going to be sexual reproduction जहाँ पे gametes form होने हैं for the production of gametes. So wherever there is gamete production, generally in sexually reproducing organisms, there is going to be meiosis occurring in the cells. So that is the main meaning about the third point about meiosis. So as we saw that meiosis is going to be a double division. So दो divisions जो होते हैं, it is been named as meiosis 1 and meiosis 2. Meiosis 1 जो होता है, it is going to be a reductional division, and this is where the actual meiosis occurs. That is, the division of chromosome is going to result in half. So जो diploid से haploid होना होता है, that is going to occur in this particular phase. And then finally, to keep the chromosomes perfectly divided, the second division will occur, which is going to be called as a equational division. And in here वहाँ पे DNA replication नहीं होगा. So here there is no DNA replication, only there is going to be the division of the nucleus. और ये division of nucleus होगा wherein the number of chromosomes are going to remain the same as they were produced in the meiosis 1. So meiosis 1 में क्या हुआ था? It was a haploid condition. So haploid condition के ही finally divide करके there are going to be daughter cells formed. So if you see here in the diagram as well, जो cell interphase complete करके आया है into the mitotic phase, there are going to be homologous chromosomes which will undergo meiosis 1 and then meiosis 2, resulting into 4 daughter nuclei which will form the daughter cells. So ये था introduction about the meiosis having the two divisions. So let's now understand each of these division in detail. So वहाँ पे जैसे we saw about meiosis 1. So meiosis 1 का जो first phase होता है, it is going to be called as a prophase 1. So if you have seen the videos about mitosis, you very well know that what is prophase and the rest of the phases that will follow it. So अगर आपने नहीं देखा है, then you can go and click on the part 2 of this particular video. Link will be provided in the description box down below. So you can surely go and check it out so that you do not feel any new things studying over here. So prophase जो है, it is going to be a much more elaborate procedure as compared to that of the prophase of mitosis. So अभी आपको ये तभी समझ आएगा when you know about the prophase of mitosis. So you go and understand the prophase of mitosis if you have not, and then if you have, then you very well know that how is it going to be much more elaborate. So elaborate है because it is going to be divided into further 5 stages. So पहले तो अपने पास जो मेटाफेज होता था, उसके बाद we used to have what? We used to have prophase, then we used to have metaphase, then anaphase, then the telophase. So this was about the karyokinesis, right? उसके बाद होता था cytokinesis. But now what is going to happen in this also? You are going to have the prophase. Prophase के भी you are going to have 5 divisions. So because of that, now you have how many steps? You are going to have the 5 over here, 6, 7, 8 and then 9th of the cytokinesis. ये हो गया only for meiosis 1. Right, meiosis 1 हो गया. वैसे ही same condition is also going to occur for meiosis 2. So meiosis 1 के 9 steps, meiosis 2 के 9 steps, that is going to give you the entire meiosis procedure. So that is the reason we have to understand it in much more detail than compared to the mitosis. So prophase 1 जो है, उसके 5 stages जो होते हैं, it is going to be leptin, zygote, paclitine, diplotine and diakinesis. So these 5, इनका order याद रखना बहुत important है. So order is going to be important, what comes after each other. So this can be asked as a MCQ question. ये प्रोफेज का लेप्टोटीन, ज़ाइगोटीन, पैक्लिटीन, डिप्लोटीन एंड डायकाइनेसिस को करेक्ट ऑर्डर में you have to arrange them. So for that you need to remember them as it is, or you can also form some mnemonics or tricks so that you can remember the particular order. So let's go through these different stages in detail कि ताकि हमें पता चले what changes are going to occur in this nucleus. So first is going to be the leptotene. Leptotene क्या है? Leptotene is a part of prophase 1. So you should not forget about it. Leptotene, what is it? It is a part of prophase 1, right? So leptotene जो होता है, in this particular phase of prophase 1, you are going to have the condensation and coiling of the chromatin. जैसे हमने आपको पहले माइटोसिस में भी बताया था that these chromatin fibers are going to be very moving out here and there. It is very difficult to divide into different groups. So उसकी वजह से condensation होगा and there will be chromosome formation which will be much more easier to handle. So for that reason condensation and coiling will start in this leptotene, and then finally when the chromatin material is going to condense, it will form distinct chromosomes and they will be visible under the light microscope. So leptotene में major event क्या हो रहा है? Finally they are going to form the distinct chromosomes by the process of condensation and coiling of the chromatin fibers. So ऐसा कुछ दिखेगा cell about the prophase 1. Next is going to be about the zygote, which is again a part of prophase 1, but the second part of it. So जैसे ही condensed chromosomes form हो चुके हैं, these chromosomes now are going to be 2 in number, because it is going to be what kind of cell? It is going to be a diploid cell. और diploid cell को आप कैसे represent करते हो? You are going to represent it by 2n condition, right? So it is a diploid cell which is going to have 2 sets of chromosome, और वो दो set के chromosomes होते हैं, you call them as homologous chromosome. So homologous जो chromosomes होते हैं, these are going to come from your mother, और एक पेयर जो आएगा it is going to come from your father. So जब zygote formation होता है, when the fusion is going to occur, so if you know about the gamete formation, गैमेट फॉर्मेशन में एक साइड से एग आया विद अ हैप्लॉइड कंडीशन, एक साइड से स्पर्म आया विद अ हैप्लॉइड कंडीशन, एंड दे बोथ आर गोइंग टू फ्यूज, एंड व्हेन दे फ्यूज व्हाट इज गोइंग टू हैपन? A zygote will be formed which is going to be 2n in condition. So ये 2n condition कैसे form हुई? One was because of the mother's genes which are present in it over here, और एक साइड से जो आया that was coming from your father which is also in it. So this is going to make the particular diploid condition, and these 2 sets of chromosome, one coming from mother, one coming from father through the gametes, is going to make up the homologous chromosomes. अब ये homologous जो chromosomes होते हैं वो अपने आप को pair करना शुरू करते हैं, they come closer to each other. So suppose this is your one particular chromosome coming from the mother's set and the other the father's set, they both will start to come together. So ये जो है this is going to be called entirely as one homologous chromosome. ये क्या है? This is one pair of homologous chromosome. वैसे ही और pairs होंगे, and this will continue up to how much? This will continue up to 23 pairs of chromosome. Up to what? In the case of human, right? Human के केस में 23 pairs होंगे, that is going to make up 46 chromosomes. So ये क्या है? एक आया mother से, एक आया father से, both are having same type of genes, they will combine together and form a chromologous pair. So now here all these homologous chromosomes are going to come onto one side and they will try to pair in side by side, which is called as synapse. So synapse क्या होता है? Synapse is going to be once they come together and form a pair, this pair is going to be called as a bivalent. उसे क्या बोलते हो? Bivalent. जैसे यहाँ पे I tried to create a box to understand for you that these are going to be together, that is nothing but bivalent. वो साथ साथ में चिपके हुए रहेंगे एक दूसरे से, that is going to make them as a bivalent. So there is going to be a chromosomal synapse that is going to occur because of a synaptonymal complex, and that will help them to stabilize throughout the process, being a two homologous chromosome. So finally crossing hone tak you have to wait, and these will be stuck together to each other by forming a synapse. और synapse form हो चुका है, that means they have been glued together, वो दोनों एक दूसरे से चिपके हुए हैं. So once they are glued together, these will be called as what? They have a new name that is called as bivalent. So if you can understand over here that this one box is nothing but going to be called as one bivalent. What is it going to be called as? One bivalent. So two homologous chromosomes are going to form one bivalent. अब अगर आपने यहाँ पे देखा है about the diagram, it also states that these are going to be having 4 homologous chromatids or 2 homologous chromatids, right? So that was about the phase called as zygote. नेक्स्ट इज अबाउट पैक्लिटीन. पैक्लिटीन जो है, जैसे ही ग्लूड अप कंडीशन में द क्रोमोसोम्स आर गोइंग टू कम, दीज आर गोइंग टू अगेन कंटिन्यू टू बिकम थिकर एंड शॉर्टर. वो बहुत ज़्यादा कंडेंस होना शुरू हो जाएगा बिकॉज मेजर पार्ट ऑफ द कंडेंसेशन इज गोइंग टू गेट completed in the part of prophase, and as you remember that Paclitine is a part of prophase 1. So you should not forget about it that still we are studying only the prophase 1. So वो अभी भी condense करते रहेंगे, they will become thick and short, and then over here a major event is going to occur that is called as crossing over. So crossing over क्या होता है? Crossing over is going to be, one chromatid is going to come over the other. और ये कौन से chromatids होंगे? These are going to be between the non-sister chromatids and between the homologous chromosome. So this particular line is super important because यहाँ से बहुत सारे questions आते हैं, and it is very important to understand that it is a non-sister chromatid but a homologous chromosome. तो आपको कैसे घुमाएंगे? How they will confuse you? They will add sister chromatids and non-homologous chromosomes. They might add you non-sister chromatids, non-homologous chromosomes. So इसे बहुत सारे options create कर सकते हैं. But what you need to remember that the crossing over is going to occur between non-sister chromatids of the homologous chromosome. That is all that you need to remember. आपने ये याद करके रखा है, that as soon as you read the options you will get your answer, and with that you have a +4 mark. So that is all done. Then how is this crossing over actually going to occur? So एक homologous chromosome को we will try to understand. So for example, this is your mother's pair of chromosome. ये आपका आया maternal side से, and the paternal side chromosome is also present, and these are going to be in a bivalent condition, right? अब ये तो है, these are a bit far away, but once the synapse is going to occur, they will be very close to each other. And now what will happen? You need to understand that what is a sister chromatid, what is a non-sister chromatid. अब आपको ये बात तो समझ आ गई that these two particular chromosomes are going to be homologous chromosomes. So I don't think that you have any problem understanding that these are homologous chromosomes. These are two homologous chromosomes coming from the two different gametes, right? Now what you need to understand is which is a sister chromatid and which is non-sister chromatid. So if you want to understand what is a sister chromatid, so ये जो red दो है, the two red are going to be nothing but called as a sister chromatid. Why sister chromatid? Because वो एक ही chromosome के है. These are just a part of a single chromosome. So both are in red color, so these are sister chromatids. वैसे ही in the case of blue, you will also have sister chromatids. So same color, that is sister chromatids. But if you take the different color, आपने एक red लिया, एक blue लिया, that is going to make the non-sister chromatids. So I hope you understood what is the meaning of homologous chromosomes, what are sister chromatids and what are non-sister chromatids. So same color से denote है, that means these are sister chromatids. So part of a single chromosome are called as sister chromatids, and part of two different chromosomes of a homologous chromosome are called as non-sister chromatids. So अब non-sister chromatids कौन से होते हैं of homologous chromosome is understood. So the crossing over will be between this particular red part, and the crossing over from the other one is this. So in दोनों में crossing over कैसे होगी? It is going to occur in this particular way. So this is one strand, second strand यहाँ पे आया, this is going to get darker because this is going to be the non-sister chromatid. और अभी यहाँ पे overlapping होगी, and this overlapping is going to be nothing but called as a crossing over. So यहाँ पे क्या हुआ है? यहाँ पे हुआ है crossing over. So I hope with the diagram it is much more easier for you to understand and how is it going to occur. So ये crossing over जो हो चुका है, this is going to lead to a formation of recombination, because यहाँ पे क्या हुआ है? यहाँ पे the genes have overlapped, the genes are exchanged in a way, and because of the exchange of genes there is going to be a recombination in the genetic material, and the genetic material recombination की वजह से there is going to be a lot more changes that can be produced in the meiosis. So whatever is the site of this crossing over, यहाँ पे exactly कैसे crossing एक दूसरे के ऊपर नहीं आएंगे, these are going to be having a enzyme reaction. This enzyme mediated process is going to occur because of an enzyme that is called as recombinase. So recombinase एक enzyme है which is going to facilitate this crossing over, and finally because of the recombination there are going to be chromosomes or further chromatids that are going to be linked at the site of the crossing over. So that was about how the actual crossing over happens. So अब यहाँ पे जो crossing over हो चुका है, now these are going to be called as a tetrad. What are these called as? These are called as tetrad, and this tetrad is going to be, as a equational form आपको पूछा जा सकता है that one tetrad is going to have what? So you can answer to them as one tetrad is going to consist of 4 homologous chromatids. So this is a very important point to understand कि एक tetrad में exactly क्या क्या चीजें होती है. So one tetrad will have 4 homologous chromatids, and if other in a way if they ask you, then you are going to say that these are going to have 2 homologous chromosomes. So 2 homologous chromosomes, or you can say that 4 homologous chromatids are going to form what? A one tetrad. So ये जो हमने दिखाया यहाँ पे, this is going to be one tetrad that you need to understand. So that was all about the stage called as Paclitine. The next stage over here we come to is diplotin. Diplotin में there is going to be completion of the crossing over, and finally the 2 homologous chromosomes will start to separate from each other. अब ये separation जो होगा, it is going to be sliding off over each other. वो डायरेक्टली खींच के आप सेपरेट नहीं करोगे, देयर विल बी अ स्लाइडिंग मैकेनिज्म दैट इज गोइंग टू occur. So sliding mechanism से they will start to slowly and steadily separate from each other, but because of the synaptonymal complex, यानी exactly जहाँ पे crossing over हुआ है वहाँ पे because of the crossing over that place is going to be still intact over each other. So synaptonymal complex will dissolve and only the region of crossing over will be kept attached, and that attachment is going to be nothing but called as a chiasmata. So whatever is the point of attachment जहाँ पे crossing over हुई है, after the dissolution of synaptonymal complex, that part will be called as chiasmata. So यहाँ पे you can understand with the help of a diagram how a chiasmata is going to look like. So that particular crossing over, so you can see a chiasmata forming over here, you can have a chiasmata here as well as one over here. So एक ही chromatid में you can have many more chiasmata formation डिपेंडिंग अपॉन कि कितना लंबा क्रोमोसोम था and how much of the crossing over has taken place. So based on that you can see the number of chiasmata varying. So over here you need to understand that the homologous chromosomes will start to separate, but at chiasmata वो separate नहीं हुए हैं, and because they have not separated at this particular point they will have a shape that is called as X-shaped structure. So chiasmata formation जहाँ पे भी होता है वहाँ पे X-shaped structure form हो जाता है, and this X-shaped structure I had already drawn in the previous one as well, and this will be like this. यहाँ पे separation होना शुरू हो चुका है. For the sake of understanding let us show this arm a little longer. ज़रूरी नहीं है that this is longer, just for your understanding let us show it a bit longer, right? So यहाँ पे ये arm लंबा हो गया है to understand purpose only. So whatever is going to be seen over here, this part is a X-shaped structure, right? This S-shaped structure is nothing but the chiasmata formation. So I hope you understood about over here the diplotin phase, जहाँ पे chiasmata formation is very important for the phase recognition and as well as the dissolution of the synaptonymal complex. The next and the last phase of prophase 1 is going to be about diakinesis. Diakinesis जो होता है वहाँ पे spindle assembly start हो जाएगी because it is going to be the last phase of prophase 1. So this is last of prophase 1, जिसके बाद the metaphase is going to come, and because of that the spindle assembly should start, and these are going to help in the separation of the homologous chromosomes in formation of two cells. So ये होगा in the case of cytoplasm. So ये changes कहाँ हो रहे हैं? These are going to occur in the cytoplasm, but now the terminalization of chiasmata is going to occur in the nucleus region. So ये nucleus के related है changes, and these are going to be related to the cytoplasm. So finally the terminalization of chiasmata will occur. Chiasmata क्या था? The X-shaped structure that was seen because of the crossing over, and this crossing over is leading to the genetic variations. So chiasmata अब dissolve हो जाएगा, disappear हो जाएगा with a theory that is called as slipping off or sliding, and once the chiasmata has been removed, what are you going to see? You are going to see the recombinant chromatids. So recombinant chromatids कैसे दिखेंगे? You can very well see in this particular diagram as well कि उनका जो red and blue color mechanism से I had told you or I have explained you. So these are going to look different. वो कैसे different लगेंगे? Because one part is going to have a blue area and the other one which was completely blue previously, अब उसके पास थोड़ा red part भी आ जाएगा. So that is why, because it is going to be crossing over. So crossing over में ये red part came to the blue one and the blue half came to the red one. So that is about the recombinant chromatids. So now these recombinant chromatids are going to go further into the metaphase 1. So finally now the nucleolus will disappear, nuclear envelope will disintegrate, and all these chromosomes are going to be led free into the entire cell. अब मेटाफेज 1 में जैसे हमने माइटोसिस में पढ़ा था that in the metaphase all the chromosomes will start to align themselves on an equatorial plate, but यहाँ पे कौन अपने आप को अरेंज करेगा? The arrangement will be of the bivalent chromosomes. So I had told you what are bivalent chromosomes. Bivalent chromosomes are nothing but the tetrads, which are going to be the homologous chromosomes. So these bivalent chromosomes are going to be coming together on the equatorial plane, and then these will arrange themselves in two rows. Why two rows? Because यहाँ पे the homologous chromosomes are going to start pairing up. So homologous chromosomes pair हो रहे हैं. That means one side you will have the red ones sitting with the recombination, and other side you will be having the blue ones sitting. So this will be combinedly sitting on one plate. So एक axis यहाँ पे होगा, second axis center में यहाँ पे होगा, and because of these two you will be calling it as a double equatorial or metaphasic plate. So finally the chromosomes will be placed on either of the poles. There is no fixed pattern कि एक ही साइड पे पैटर्नल होंगे या एक ही साइड पे मैटरनल होंगे. As you can see up here in the diagram, यहाँ पे position कैसी है और यहाँ पे कैसी है. So it is no fixed pattern कि all the maternal ones will be sitting on a single side of the plate, but randomly they will be placed onto the metaphasal plate. And now जैसे मेटाफेज ऑफ द माइटोसिस में भी हमने देखा था that the microtubules will come and they will attach to the centromere, और centromere पे जैसे ही attach करेंगे they are going to shorten into length, that is leading to the next phase that is going to be called as Anaphase 1. Anaphase 1 में since the chromatids have been attached to the microtubules of the spindle fibers, now the shortening of the microfibers will occur, जिसकी वजह से the two homologous chromosomes are going to separate from each other. So it is very important because हमने ये भी पढ़ना है, that is called as Anaphase 2. So Anaphase 2 जो हम पढ़ेंगे वहाँ पे you need to also observe that in Anaphase 1 the separation is going to occur between the two homologous chromosomes, whereas in Anaphase 2 homologous chromosomes की जगह पे only a single chromosome is going to undergo separation. So Anaphase 2 में who is going to separate is going to important, who is separating, whereas Anaphase 1 में what is going to separate is going to be important to understand. So Anaphase 1 में two homologous chromosomes will separate and they will start moving towards the opposite poles. And now what is going to be the result? The result is going to be a univalent. Univalent है because homologous chromosomes में से एक पेयर तो चला गया ये साइड पे, right, and the second pair had come to the second part of the pole. So एक pole यहाँ पे है, the centromere over here and one centromere over here. So this will be the condition of the Anaphase 1. So one chromosome going here, one chromosome over here. If you compare this is going to be the diagram for Anaphase 1. If you compare this diagram to the Anaphase of mitosis, अगर आपको mitosis का याद है and you can understand or relate to it, the condition is going to be a bit different. How is it different? Because वहाँ पे only a single chromosome had different conditions. वहाँ पे if you remember, and only one chromosome was splitting into two. एक यहाँ पे attach हो रहा था, एक यहाँ पे attach हो रहा था. So if you remember, you can see how is the difference of anaphase of mitosis and the anaphase of meiosis, that is the first one. So how is the difference? यहाँ पे सिर्फ एक ही chromatid है, whereas in Anaphase 1 you are going to have two chromatids or one complete chromosome, and this one complete chromosome is nothing but going to be called as a univalent with two chromatids attached together at a centromere. So वहाँ पे centromere split नहीं होगा, which was actually occurring in the anaphase of mitosis. यहाँ पे क्या हो रहा था? Here it was splitting of the centromere. So here there is no splitting of centromere in the Anaphase 1. Anaphase 1 में there is going to be splitting of homologous chromosomes. So this is a very important concept that you need to understand. कि exactly Anaphase में who is going to split, right? Anaphase 1 में splitting of homologous chromosomes, Anaphase of mitosis as well as Anaphase 2 there will be splitting of the centromere. So finally, once all the splitting has been occurred, once they reach into the different poles, now they will start the next phase, which is going to be telophase 1. Telophase 1 में all the chromosomes have reached their respective poles. So once they have reached, now it is time to form the daughter nuclei. So nucleus will reappear, nuclear membrane reappear हो जाएगी, and then all the chromosomes will start to uncoil themselves, and after uncoiling there will be production of two daughter cells having one single nucleus. Along with it, that is one chromosome per of the homologous pair. So finally, whatever the single nucleus has been obtained of the daughter cells, this will be in the haploid condition. So very important to remember, जो टीलोफेज 1 के बाद जो न्यूक्लियस बनेंगे, those nucleus are in haploid condition, they are no longer in the diploid phase. So now जैसे ही टीलोफेज 1 complete हो जाएगा, there is going to be cytokinesis that we have already discussed in the previous video. Similarly, the cytokinesis will occur, and as soon as the cytokinesis gets completed, there is going to be a intermeotic phase. So उसके पहले क्या होता है, there is going to be a intermeotic phase, wherein in the intermeotic phase there is going to be again some preparatory period. This preparatory period will be for formation of, you can say, ATP बना लिया, where you formed some proteins. All those are going to be formed in this intermeotic phase, and after this intermeotic phase you are going to have meiosis 2. So meiosis 2 is going to be very much similar to the mitosis जो हमने part 2 में cover किया था. So mitosis is very much similar, and we are using the term that is similar. It is not an exact copy of mitosis. And what is the differences? Differences are going to be discussed in this table. So the differences between the two is that mitosis is going to occur in diploid somatic cells, whereas meiosis 2 will always occur in haploid germ cells, right? The mitosis will be always followed by a DNA replication, whereas meiosis 2 में there is no DNA replication before the division. So that is point number 2 of the difference. Third point is going to be that in mitosis, daughter cells जो होंगे, they are going to be similar to the parent cell as well as to each other, whereas meiosis, these are not at all going to be similar, neither to their parent cells nor to each other. So how is that going to be? So here are some diagrams that can help you to understand. Mitosis का जो division होगा, mitosis के बाद both like two daughter cells are formed, but still for the second mitosis as well, these are going to have same type of chromosomes. So I have represented it with pink color. So all are going to be similar to each other, whereas if considering meiosis 2, उनका जो result होगा of the daughter cells, of the 4 daughter cells, all the 4 will be different to each other. एक दूसरे से कोई भी similar नहीं होगा. So here you can see there is a pink and the green, and here also you can see a pink and the green. Here you can see only pink strand, here you can see a green strand. So all 4 are going to be very much different. That is the difference between meiosis 2 and mitosis. Now there are going to be similar phases. जैसे हमने meiosis 1 में cover किया about prophase 1, metaphase 1, anaphase 1 and telophase 1. Similarly, in the meiosis 2 as well, you are going to have the 4 phases, that is prophase 2, metaphase 2, anaphase 2 and the last, that is telophase 2. So prophase 2 जो होगा, it is not going to be as long as the prophase 1. Prophase 1 जो होता था, उसमें there were further 5 divisions that we have already studied in much more detail. So वो जो 5 divisions है, those 5 divisions are not going to be in the case of prophase 2. The prophase 2 is much more easier to understand. Just like mitosis, यहाँ पे भी chromatids जो होंगे वो condense करेंगे, और कौन से chromatids होंगे? They are going to be univalent chromatids, and now these are going to condense and become compact, and finally there will be nucleolus and nuclear membrane that will disintegrate and disappear, so that the entire cell can be used for the separation. So that was about prophase 2. Second जो आएगा, that is anaphase 2. जैसे ही कंडेंसेशन हो गया है, chromatids formation has been taken place, the univalents or the chromosomes will align themselves. So alignment होगी कहाँ पे? At the equator or the center line of the cell. And here also again the microtubules will come from the opposite poles towards the equator. They will attach at the kinetochore and they are going to start to connect to each and every chromosome और उनको एक लाइन में इक्वेटर के पास लाके खड़ा कर दिया जाएगा. So difference between metaphase 1 and metaphase 2. So here they are going to be equator which is only one in the case of metaphase 2. मेटाफेज 2 में एक ही इक्वेटर प्लेट होंगी whereas there will be double plate formation in metaphase 1. So now as you have studied both metaphase 1 and 2, you know the difference about the diagrams as well. So if you can't, then rewind the video and try to draw those and then you compare it with this diagram of the metaphase 2. So मेटाफेज 1 में कैसी अरेंजमेंट थी और मेटाफेज 2 में कैसी अरेंजमेंट है, that needs to be understood because diagrams are going to be asked as MCQs. So diagrams जो होंगे diagrams पे MCQs पूछे जा सकते हैं to identify the phase, right? They can ask you to identify the phase and then they will ask you some questions. So for identification of the phase, you need to understand the diagrams very well. So for that reason, you compare both the diagrams of metaphase 1 and 2. Similarly, आप prophase के भी कर सकते हो. You can also do it for anaphase and telophase. So let's move on with the anaphase 2, जहाँ पे जो centromere होगा which is holding the chromatids at its place. Now it will be separated. So anaphase 2 में क्या हो रहा है? Anaphase 2 में there is a splitting of centromere and because of centromere is splitting there is going to be the anaphase 2. जबकि anaphase 1 में क्या हो रहा था? Anaphase 1 had the splitting of homologous chromosome. So that is very well told to you before as well. So splitting of homologous chromosome. So not writing it completely, but now you know what it is, right? So splitting of homologous chromosomes anaphase 2, splitting of centromere anaphase 2, right? So finally, once the chromatids have reached their respective poles, the last stage will occur, that is going to be telophase 2, जहाँ पे chromatids होंगे, they will start to uncoil themselves, they will decondense, become thin, and finally there are going to be 4 haploid nucleus formed. So 4 haploid nucleus, you can see them very well how these are going to be. And यहाँ पे क्या हो गया है? Finally these are going to have recombination that is going to occur. So because of recombination that has occurred, it is going to lead to variation. And since variations are going to be created in these cells, that is going to give better survivability rate. Get better survival rate because in nature you need some or the differences that is going to make you unique from others, and that uniqueness will help you to survive in the competition. So better survival rates to undergo the nature's competition, this is going to help. Now what is the significance of meiosis? हमने पढ़ लिया completely meiosis कैसे होता है and what are the steps and differences are going to occur. But why in the nature this meiosis has actually been promoted? Reason number 1 is formation of gametes, because gametes जो होते हैं these are going to be in haploid condition. So haploid condition में gametes जो form होंगे, as I always tell you that this is going to be the egg which is going to be in haploid condition. So egg coming from the maternal position, the sperm which is going to come from the paternal side. Now the sperm is having one haploid set, egg having one haploid set. Once these are going to come together forming a zygote के condition में what is going to happen? The red pair is going to have how many? 23 chromosomes over here, and whereas the blue one is going to have 23 chromosomes. Okay, so red के 23 will come over here and blue के 23 will come over here, and finally they are going to make what? 46 chromosomes found in the case of humans. So ये 46 continue करने के लिए you need to have 23 coming from the egg, 23 coming from the sperm, so that the zygote can have 46 that their parents already have. So यही same concept use होता है that maintenance of the chromosome number maintain रहे, so that the original diploid phase can be maintained. For that reason meiosis is going to help. And then finally a very important change that occurs because of meiosis, that is introduction of variations. Since there is going to be recombinations की वजह से ही there is going to be variations, and once the variations have been introduced into the genetics, there is going to be better survival rate, or survival rate होगा better, then and then only there is going to be the species that can survive in the nature's competition. So that is the significance of meiosis. So we have completed the entire theory about meiosis. Meiosis की theory के ऊपर अगर कोई भी question आ जाए, I hope that you can solve all the questions regarding this video lecture. You can also solve the numerical questions. So as we are going to discuss over here, यहाँ पे दो type के questions पूछे जा सकते हैं. The first one being that you ask the number of meiotic divisions that are required to form a certain number of pollen grains. तो यहाँ पे we have the question for 12 pollen grains. So first of all, as reading the question, you should remember that pollen grains जो होते हैं, these are going to be nothing but the male gametes of the plants. So यहाँ पे आपकी plant knowledge भी test होगी, because until and unless you don't understand that pollen grains are male gametes of the plants, you will not understand कि gamete formation में there is going to be requirement of meiosis. So meiosis में क्या होगा, there is going to be one pollen mother cell यानी जिसके अंदर there is going to be meiosis, and one pollen mother cell when undergoes one meiosis, there is going to be formation of 4 pollen grains, right? So it is very important to understand one meiosis gives you 4 pollen grains. So if you want to have x number of pollen grains, how many meiosis do you need? So you are going to get it by a simple formula, that is the number of meiotic divisions that are required to form x number of pollen grains. तो यहाँ पे x की वैल्यू कितनी है? x की वैल्यू you are going to have 12. So 12 को डिवाइड करो 4 से, that is the formula, you will get the number of meiotic divisions required. So that is one number type of question. Second type can be asked to you, that is how many meiotic divisions are required to form 12 fruits. Now here you need to remember यहाँ पे 12 fruits है, whereas here you have been given that is 12 pollen grains. So this is the difference that you need to remember. Pollen grains जो है वो male gametes है, whereas fruits, these fruits are coming from where? ये fruits होंगे after fertilization. Very important to understand. ये fertilization के बाद होगा, and once the fertilization is occur, that is fusion of gametes has occurred. So fusion of gametes तब होगा जब gamete formation होगा. So fusion of gametes के लिए you need to also prepare gametes. So preparation of gametes में क्या लगेगा? Preparation of gametes में you are going to require, that is going to be meiosis. So now to prepare both the gametes, so gametes में कौन आएगा? You will have the female gamete as well as you will have the male gamete. अब फीमेल गैमेट जो होगा, फीमेल गैमेट के meiosis में you need to remember that one meiosis is going to give you how many eggs? One meiosis will give you one egg, whereas in the case of male gametes, that is the pollen grains in here for the case of fruits. So one meiosis is going to lend you how many pollen grains? Pollen grains मिलेंगे 4. Okay, so that is the condition that you need to understand as soon as you read the question. So 12 fruits है, fruits कब बनेंगे? That is only after fertilization. So fertilization हो रहा है, that means there is fusion of gametes. As there is fusion of gametes, you need to prepare the gametes first so that the fusion can occur. So एक बार gametes बनाने हैं, that means you need to undergo meiosis, which will occur in the female as well as in the male part of the plant. So once you know this particular thing, then and then only you can solve the further of the question that how many meiotic divisions are required. So iske liye bhi you have a very simple formula, but this why did I explain you? So that you just don't remember the formula and solve the question. आपको ये भी पता होना चाहिए that how that formula has been derived, so that by any chance if you don't remember the formula you can understand the concept behind it and can solve the questions as needed. So यहाँ पे एक meiosis एक egg बनाएगा, एक meiosis 4 pollen grain बनाएगा, and then finally how much do you need? You need 12 fruits. So 12 fruits है, यानी 12 fruits में you need 12 female gametes as well. So 12 female gametes चाहिए, that means you need 12 eggs, and when you need 12 eggs, what do you mean? You need 12 meiosis. So एक साइड पे आपको कितने meiosis चाहिए? एक साइड पे आपको चाहिए 12 meiosis. And for the case of male gamete, how do you need? आपके पास 4 pollen grain मिल गए from one meiosis, and here how many more do you need for formation of 12 fruits? For fertilization of 12 eggs you need 12 pollen grains. So that is going to give you more 3 meiotic divisions. So finally combining them both, that is 12 plus 3, you will get your answer as 15. So इसका answer क्या आ जाएगा? You will need 15 meiotic divisions for formation of 12 fruits. So यहाँ पे simple formula आ गया. So let me erase this thing so that you can read the formula over here. So whatever is going to be the requirement of to make x number of fruits, x number of fruits will be forming from how many meiotic divisions? From x plus x by 4 divisions. So अगर आपको fruits की जगह पे seeds पूछ ले या grains पूछ ले, या even if you get the same type of question with fruits, how many meiotic divisions are required, you can use this formula and you will get the correct answers for the number of meiotic divisions. So I hope you understood this different type of numerical questions that can be asked to you in the exam, and with this you can correctly understand the concept, और formula से quickness के साथ भी you can solve the question. So that was all from my side for this chapter. So we have completed the chapter about cell cycle and cell division. अगर आपको इससे कोई भी help मिली हो, and if you feel that others should also get some help, then don't forget to like, share and subscribe to our channel, that is Biophilia, and you can surely go and check out some other chapters of our chapters on this channel, and we will be welcoming your comments in the comments section down below. आपको अगला कौन सा chapter चाहिए या or what sort of videos do you want, you can comment down in the section below, and you can connect to us sharing your doubts or other things that you could then tell. We will meet back soon once again with a new chapter. Till then, keep smiling, keep studying, work hard. Bye bye everyone. Thank you.